Hey, hello everyone. This is Prashant, and now we are going to cover a new unit known as ionic mechanisms of cell or a neuron that we are discussing. So, in the previous lectures, we discussed neuron reside within nerves, they conduct information via electricity, and they may have some form of chemical nature that we are going to discuss right now. So, let me just start with my PPT and we can move forward. So, yep, okay, I'm going to go into the presenter view. And yeah, let's go. So basically, we are borrowing certain concepts from different fields and we are going to integrate it in our study of neurons because without them, we cannot go further ahead. And the main two fields we are going to cover are physics and chemistry. And the first, what you call it, principle or phenomena we are going to borrow from physics is electrostatic attraction. So basically, opposite charges attract each other, similar charges oppose each other, observable on a physical level. And we also know, so positive repels positive and negative repels negative, positive attracts positive. But no, positive attracts negative. Okay, cool. That was a little bit of a slip there. And then we borrow from the atomic theory. What that charges are properties of things. Things what kind of things? That are ions of the atoms. Atom, most fundamental unit we have. An atom has electrons, neutrons, and protons. And the change in the composition of electrons causes charges. Okay, because electrons are the most free moving subatomic particle of an atom. Okay, cool. So neurons have positive ions. The following ions are positive ions are present, known as cations, are sodium, potassium, calcium, and there are negatively charged ions known as anions, and they are chloride ions, protein ions, represented by A minus. And we also know that electric current is due to the flow of charges from positive to negative. Cool. And then another concept we have is a property of gases. Generally, ions move from high concentration to low concentration regions. So, just like a gas spreads all across the room, similarly, ions of the gas also spread across the room. And ions can also spread across water as well. As long as they are free moving, they will move. Okay. So when the concentration of ions is equal on both the sides, for example, concentration of Na plus or Cl minus is equivalent across the membrane, then we can say that the uh, the charges have equalized or in a state of equilibrium. Now there is no movement of charges, everything is kind of stable. So that state is known as equilibrium. And now let's start with our first scientist who did research on the ionic mechanisms of neuron. Okay, how chemical or ions affect neurons. So first we kind of have to go to the medical field, and we have our scientist whose name is Sidney Ringer. Okay, so what did he did? Okay, so his method was as follows. So he isolated a frog's heart and then put it in a perfusion apparatus. Basically, it's artificially beating a heart by injected saline solution in it. And he knew the composition of blood by chemical methods, that blood is made up of different forms of salt, for example, NaCl, KCl, and CaCl, sodium, potassium, and calcium, and chloride. Those are the four main elements in your blood. And then he found out that when you change the concentration of these ions inside that saline solution, it affected their heartbeat. How it affected them, we are going to find out. So let's go with his reasoning based on his observations. So he found out that heart rate is affected by ion concentration. Okay, and what were those ions? Sodium, potassium, calcium, chloride, and there must be a balanced concentration for a heart to function. Okay, so that was called Ringer's solution. That is kind of used in labs as well nowadays. And then he found out that those four ions are essential for the functioning of the heart. Okay. So what kind of logical deductions he made? He first found out that heart supplies blood to brain and to all other body parts through arteries that is anatomically observable or okay. Then we know that brain consists of neuron cells from staining and stuff like that. And from neuron theory, we know that if a neuron is a cell, it will also have certain similarities with a cell. Its differentiator is that it transfers information, but it also contains the part of the cells like cell body, uh, cell membrane, nucleus, etc., etc. Okay. So we know that okay, neurons have proteins and they get their nutrients from the blood. So and neurons transmit electricity or electrical signals. So electricity occurs due to the movement of charge. We also know that. And combining all these information, we can come to the conclusion that ions, specifically ions of sodium, potassium, calcium, and chloride, alongside negative protein ions, must play an important role in neural transmission. And now we are going to find out how do they play the role, and if they play a role, we are going to verify that too. So we are starting with our next scientist, whose name is Julius Bernstein. So his main work was giving a hypothesis on a cell membrane, specifically that of neuron. Okay, so obviously from cell theory, by knowing the nature of a cell that was discovered by Hooke and others, we came up to a certain findings that a cell has a membrane, it separates it from its surrounding. It's semi-permeable, so nutrients can go in and out. Okay, then there are certain form of pores or gates because it's semi-permeable. There must be some openings, some form of openings that allow some what you call it nutrients or some chemicals to go in and some chemicals to go out. Okay, that is logical enough. Now we move forward. Uh, to our next step in reasoning, that is ions exist in cellular fluids, because obviously under microscope we can see that most of the human body consist of cells, and cells consist of reside in a fluid medium. And so yeah, the ions must reside in a fluid medium because neurons may use ions. And then he found out that there is a difference in concentration. For example, inside the cell there will be a difference of ions, and outside the cell there will be a difference of ions. Okay, so it leads to a certain form of potential difference that we actually verified through previous experiment, that the charge of inside the neuron is minus 70 millivolts. Okay, and we also know that electric neurons conduct electricity. Electricity occurs due to the movement of charges, and charges are due to ions. So ions are responsible for electrical conduction in neurons. Okay, cool. And so he found out that the change in ions across a cell membrane must cause change in cell's potential, and ultimately cause is the cause for neural transmission or action potential, and that was known as membrane hypothesis. And he also found out through various chemical techniques that there are more concentration of potassium ions inside the cell than outside it. Okay, cool. So these are the conclusions we have. So we found out that okay, ions play a role in neurons, and ions of potassium so okay, potassium, sodium, chloride, negative, calcium, and negative chloride uh, have a role in what you call it neural conduction. So now we are gonna move forward, and we are come back to our two main scientists, that are Huxley and Hodgkin. And this was their experimental setup. Basically, they put electrodes inside the axon of the squid, and now they started changing the concentration of the solution. Okay, so in their voltage clamp setup, what they did was it was a certain form of circuit that brought the cell back to its action resting state after there is some form of electrical signal was stimulated or change of concentration of solution was done. So they stimulated axon in different concentration solutions. Okay, so there were different ions, and they found out that okay, suppose if we put axon inside a chlorine solution, which means it has no sodium, then there is an effect on the current generated or the action potential generated. Basically, without sodium, no action potential is generated generally, or it is dampened. So that was their observation. Okay. So what were their findings and what did they find out? Okay, their findings are can be summarized as follows: that when they isolated and noted the effects of sodium, there was some conductance. When they potassium, it also led to certain form of action potential, maybe less than sodium. But combining both sodium and potassium, there was a actual action potential that came out. They measured through ionic currents. So let's go through their reasoning. How did they figure it out? So this was their reasoning. Okay, this is a big form of reasoning, so follow me up on that. So neuron, like blood, has ample amount of ions that are present. The ions we have discussed. Ion movement causes a neuron current. Cool. And current and voltage are related. That is due to Ohm's law and discovery of other physicists. And so the change in ion movements across the cell membrane must be cause of ionic currents and action potential. Or okay, cool. And the major effect is of sodium and potassium because that's what they measured properly. And they found out that across the axon, sodium and potassium ions are responsible for ionic currents or action potential. Okay, cool. And then we found out that resting potential is an state of equilibrium. Cool, that we know that from Nernst experiment. And we found out that at resting potential, there are more negative ions inside the neuron than positive ions. Cool, that we have verified experimentally. And so they what they thought that during depolarization, when the neuron uh, when the potential inside the cell becomes less negative and moves towards positivity, more positive ions must enter the cell through the pores. Makes logical sense. As at resting, it is negative. So how will a negative thing become more positive? By uh, influx of more positive ions, obviously. In repolarization, it must again become negative. So either the negative comes in the cell or the positive leaves out. Both are the possibilities. So the positive leaves out of the cell. Okay, cool. And that they have verified experimentally through the concentration of sodium and potassium that we have seen in the graphs previously. And there must be a time difference. And we have observed that there is a time difference which causes a refractory period during which no amount of stimulation actually causes action potential. Yep. So sodium is entering the cell and leaving cell faster than potassium. And how do we know that? We know that through the graph as follows: that sodium comes faster and leaves faster, and the potassium comes later and leaves later. Okay, cool. That we can infer from the graph and the ionic, what do you call it, calculations. Okay, cool. So now let's move forward to our next scientist. So they were Arvind Nehar and Bert Sakman. Okay. So their main contribution was creating a patch clamp technique, and these are as follows. So in their method, what they did was they placed an electrode inside a glass pipette or a tube, a very narrow tube of glass, and they put an electrode in it and they filled it with saline solution so that it could conduct electricity. And the tube was so narrow that it could actually attach to specific cell part. And what they did was they measured the current generated by ions by changing the concentration of solution uh, in which the cell was under the microscope. So that's what they did. And they also passed the signals through the cell and they observed like, okay, what was the change in the ionic current as through the electrode or the micro electrode in the pipette. Okay, that's what they did. And this helped to prove the existence of individual ionic channels, that there are ionic channels that exist across the cell membrane, especially on neuron, and they were able to verify it. And they also observed what was the effect of chemicals and their mechanism. So their reasoning was as follows: change in ion concentration causes current because there is a change in potential across the cell, and we have verified that. Okay, cool. And ions move across the cell through ion channels. We know that from cell theory as well. So ionic currents are change across a single ion channel, and they help me calculate the concentration of ions. Because if I know the charge of an individual element or ion, and I know how much total charge is there, I can calculate how many numbers of ions are in a solution. Yep, makes sense. Okay, and we use a certain equation, that is Goldman equation, to know if there is a certain charge on one side of the cell, then how much charge will be on the other side. Cool. So they measured the current for each individual ion Na, K plus, Ca2 plus, Cl minus, A minus, and they mapped out what was the ionic mechanisms across the timeline, across the action potential. And we found out that certain ion channels allowed uh, or permeable to sodium or potassium, and some were gated, some were opened when it was electrically stimulated. And there was a certain mechanism called pump, which used to balance the concentration because a cell wants to maintain its homeostatic state of osmosis. Otherwise, the cell will die. So, cell tries to maintain that state. So, based on all those calculations, we found out that okay, there is more concentration of sodium outside the cell, more concentration of potassium inside the cell, more concentration of chloride outside the cell, and stuff like that. And there is more concentration of what you call it, protein inside the cell. So, simplifying the diagram, we can see that okay, protein and potassium are more inside the cell, and chloride and sodium are more outside the cell. Cool. So, this is a homeostatic state of a cell. Cool. So now we are gonna discuss the each stage of action potential in terms of ions. So let's start with our resting potential state, and this is the state. At resting potential, everything is balanced. There's the equilibrium, and in that equilibrium, diffusion has stopped because it has been balanced by electrostatic charges. More potassium ions cannot go outside the cell. Why? Because they are attracted by the negative protein inside the cell. Meanwhile, uh, the new potassium ions that wants or sodium ion that wants to enter the cell due to their low concentration cannot. Why? Because they are repelled by the positive potassium ions. Cool. And negative ions cannot move outside the cell due to their negative charge, and they also repel chloride ions. Cool. This is the resting potential state. Cool. And we know that there are three forms of channel. For example, potassium is free to enter or leave the cell. Sodium is allowed to enter the cell under certain circumstances. For example, like when the cell is electrically stimulated, the gates open and they allow the entering of sodium. Cool. And there is a pump, which is a balancing, what do you call it, balancing mechanism, which plucks out three Na plus ions and sucks in two positive ions, potassium ions. Cool. So this is the mechanism, and this pump is known as NAK plus ATP pump, and the gate of Na plus ions is generally closed, and this helps to maintain the negative charge inside the cell. Okay, cool. Now we move forward to our next stage, that is depolarization. Okay, so when there is any form of electrical signal or stimulus to the neuron, the Na plus gates open up, and due to the high concentration of Na plus ions outside the cell and negative charge inside the cell, all the Na plus ions just move inside the cell very fast, and it will lead to the rise in the uh, potential of the cell, in which the negative potential decreases due to the influx of the positive ions. Cool. And the next stage is repolarization. Basically, what happens is that now that the cell has kind of went into an extreme state, now it will try to get back to its resting potential for homeostasis. So, what you will try to do is that uh, more positive ions will be flushed out. For example, K plus ions will be flushed out. The gates of Na plus ions will be closed. Okay, and the chloride will also enter the cell. This will lead to the potential to decrease. So repolarization, it goes back to the negative. Okay. So yeah, that's what we discussed. And now we go to the hyperpolarization. Okay, what happens here is uh, uh, that potassium gates, basically, they take more time to open up and more time to close. This causes more positive uh, potassium ions to go outside of cell. This makes the overall cell more negative in nature. So it makes it more negative than resting potential, that is minus 70 millivolts. It makes it more negative. And what this causes is that it causes uh, the cell uh, potential to drop to more negative. Okay, cool. So that's what's happening because the potassium gets uh, open closed later. It causes more potassium ions to go outside the cell and make the cell more negative. And that is just the nature of the cell. Cool. And now we enter to the state of uh, depolarization again. But in this case, it's more of a state of attaining homeostasis. There is no stimulus. The cell is automatically trying to achieve its resting potential. And how it does that? The pump plays the major role. Basically, a pump also throws out all the positive ions, what you call it, Na plus ions outside the cell and allows the entry of 2K plus ions. And also over time, the gates of potassium ions also open up because of there is a less concentration of potassium ions. So the cell becomes more negative. It is starts to attract potassium ions again. So it starts to become more positive. And until the equilibrium is reached and the cell has again achieved its resting potential. So yeah, that's what happens. That's the whole cycle of action potential across the neuron in terms of ions. Cool. So overall, this is what happens. One cell action potential on one side or the charge difference on one side stimulates other gates as well. This causes a charge difference to travel across the ions, and we can measure it through what you call it our electrical instruments. And yep, and it is called as saltatory conduction. So what happens is that we know that neurons axon, especially in mammals, is surrounded by myelin sheath or insulation of glial cells that we observe through microscope. And what we found out that there is a gap between those sheets or insulations, and they have ion channels because obviously ion will not travel across cells to enter the neuron. It will enter it if it can enter directly. So this is the most direct way of ions to enter the neuron directly inside the cell membrane. Okay, cool. So they do through the gaps in also known as nodes of Ranvier. Okay, cool. So these gaps are close enough so that stimulation on one side can stimulate uh, or transfer the charge to another side without going through each channel. So some channels are covered by the myelin sheet or insulation, some are open. So the charge jumps across these gaps, and this is known as saltatory conduction. Okay, and this creates a faster level of conduction. Suppose if a cell is not insulated, what it will happen is that it has to go through each channel, and each channel will take more time, and the more time it takes the harder it will be for conduction of what do you call it, electricity. And it also defines that uh, the axons of mammals is more thinner than that of squid. So basically, we don't have that much space in our body. So it's kind of a mechanism we kind of developed, or evolutionary trait we developed, that allowed us to conduct electricity faster across our neurons. So, yep, this is how it happens. So, Na plus ions gates open up, uh, ions travel across both inside the cell and outside the cell across different channels, and the charges just jumps in between the cells, and this is how it happens. And I'm gonna show you a diagram. So yeah, the current just flows across the cell by the movement of ions between the gaps of the neuron. Yep, and action potential remains same throughout the cell. Yep. So yeah, so that's how actually the charge is travels across the neuron. And now we are going to cover our next scientist whose name is John Achilles. So what did he did? Okay, his method was trying to discover how all the neurons transfer information and how one neuron integrates that information from multiple neurons. So his method was he used a oscilloscope, that is a voltmeter in a simple language, or a form of galvanometer as well. So he used that, And what he found out that when you place an electrode across uh, different synapses, one cell is excitatory and one is inhibitory, which means one excites the motor neuron and the other is inhibits it. So he kind of measured what were the charges before that were stimulated and what was the effect on the motor neuron. So so he was discovering the potential difference or the charge in one neuron and its effect across the synaptic gap on the another neuron. And he did it for all the other neurons. Okay, cool. So he summed all the charges and he found out that there are two basic types of action potential generated. One is positive, one is negative. The positive one is known as excitatory because they cause some form of movement inside the neuron, motor neuron, and the inhibitory, which causes less movement basically inside that motor neuron. So they work in opposite direction, and that's why they're excitatory and inhibitory. And on a major level, on a what you call it, on a meta level, they work as sympathetic and parasympathetic systems. Cool. And one neuron uh, can act as inhibitory or excitatory depending on the sum of all the messages received from all the other different types of neurons. Cool. So this is what we discovered. Okay. So procedure was to put electrodes across inhibitory and excitatory neurons and measure their effect on the motor neuron in between. Cool. And this was the fact that was discovered. So he found out different combinations. The first was time. So he put a time difference between two stimulations. And he found out that earlier the stimulation, uh, you give the, it is the first signal that is received by the motor neuron as a postsynaptic potential. Okay. So, and if the stimulations are close enough in time difference, they just combine, whether they are excitatory or inhibitory. So this is the diagram. If they are different in the initial stages, they are considered as different signals. The more close enough they are, more closely they are treated as a one signal by the summation of both the signals. Cool. This is the temporal succession. There is a spatial summation in which there is a position difference. So at uh, two different stimulatory, what you call it, neurons are placed and at two different location and their effect is noted. So if both these stimulations are fired at the same time, they are considered uh, as different if they are from different locations. But if they are fired at the same location, they are treated as one. Okay. The difference is we are not taking time difference in it. We are firing at them at the same time. Okay. So at different location at the same time and at same location at the same time. So what we observe is that when the locations are different, the signals are taken as opposite. Basically, they are different. So signals can be differentiated if they are on the opposite sides. But if they are on the same side and very close to each other, then they are taken as same and they are combined. Cool. So the total summation uh is as follows. How many number of excitatory versus inhibitory signals were there from other neurons, and a singular cell combines all of those into one action potential if it is whether it's positive or negative, and that will have an effect on our uh, whatever organ this particular neuron is attached to or neurons are attached to. Cool. So this is how it happens, and that's all. And this is how electricity is conducted in a neuron in terms of chemicals, especially the ions. Okay. So, so far we have kind of briefly overviewed, okay, how ionic mechanisms travel across the neuron and between two neurons, and how one neuron combines the signals from all the other neurons. Now we are gonna uh, deep dive into what happens chemically between two neurons, specifically in that synaptic gap between those two. And that's what we are gonna discover. For example, when dendrite uh, is connected to the synapse, okay, and there is a gap, what happens, how chemicals work, that's what we are going to discover in our next unit. and I hope you liked it. And do give it a share, do subscribe if you like it, if you want to see more content like this, and do comment, and that's all.